Hello dear viewers, welcome back to another lecture. So in this lecture we are going to talk about platelets. Now before we start, let me ask you, have you ever imagined that why don't your gums bleed once you brush your teeth in the morning? Because once you brush your teeth, you create many micro punctures in the blood vessels that are present in your gums. So thanks to these platelets, what we are going to talk about today, thanks to these platelets actually seal those micropunctures we create in our blood vessels during brushing our teeth. This is just an example. Another example, if you walk, then you actually, while walking a little distance, you create many micropunctures in the blood vessels that are in the sole of your foot. So why don't you bleed there? Because again, thanks to the platelets, these platelets seal those micropunctures. Uh, Once there is the puncture in the blood vessels, these platelets seal those micropunctures within a fraction of seconds. Now, imagine that there are no platelets in our body. Without these platelets, once we brush our teeth, our gums will start bleeding and our mouth will be like a deadly crime scene. So we are going to talk about these platelets today, and our focus will be on the formation of platelets, the structure of platelets, and the functions of platelets. Let's get started. So these are the contents we are going to cover today. We will have the introduction to platelets, then we will talk about the platelet formation, that is thrombopoiesis. Then we will talk about the structure of platelets, followed by the functions of platelets, and we will conclude with the normal count of platelets and the variations in that normal count. Now, when we talk about the introduction, the platelets are actually the formed elements of blood. These platelets are not even cells. These are the fragments that are present in the blood, and these fragments are formed from the biggest cell present in the bone marrow, that is called as megakaryocyte. So these are actually the cellular fragments of the giant cell, and that cell is megakaryocyte. Now, when we talk about these platelets, so platelets, as they are the cellular fragments, they are the cell fragments, so they are anucleated. There is no nucleus present in the platelets, and these platelets are about 2 to 4 micrometers in diameter. They are 2 to 4 micrometers in diameter. So that means we can say that 2 to 3 platelets can fit in one RBC, because we know RBC average size or diameter is 7.2 microns. So we can say that based on this, that 2 or 3 platelets can fit in one RBC. Now, let's now talk about the formation of the platelets. Now, platelets are also formed from the megakaryocyte in the bone marrow, because we know that bone marrow is the site of formation of all the types of blood cells, all the types of formed elements, be it the red blood cells, be it the white blood cells, be it the platelets. So platelets are also formed from the bone marrow. Okay, now we all know that there is a stem cell present in the bone marrow. We have already covered that in the lecture of erythropoiesis. We know that there is the hematopoietic stem cell, that is the stem cell present in the bone marrow, and from the hematopoietic stem cell we have two stem cells originating from that one is called as the myeloid stem cell, and another one is called as lymphoid stem cell. Now, lymphoid stem cell give rise to lymphocytes, and this myeloid stem cell give rise to all the other types of blood cells apart from the lymphocytes. Now, from myeloid stem cell, we have after certain differentiation processes, we have something that is called as megakaryoblast. We will see in detail in the next slide. We have megakaryoblast, and megakaryoblast will form the megakaryocyte, and from the megakaryocyte we have these fragments budding off from the megakaryocyte, and those are called as platelets. Now let's talk about this in a little more detail. We have, again, I will tell you, we have the hematopoietic stem cell, that is pluripotent hemopoietic stem cell. Then from hematopoietic stem cell, we have the myeloid progenitor, that is myeloid stem cell. And from myeloid stem cell, we have the formation of different colony forming units. And in this case, the colony, we have the colony forming unit megakaryocyte. So colony forming unit megakaryocyte will give rise to the first stage technically, that is in the process of thrombopoiesis, and that stage is megakaryoblast. Now, if you remember from the lecture of erythropoiesis में हमने देखा था कि once we go from one stage to another, there are certain changes that are occurring, and one of the changes was that the cell size keep on decreasing once we go from one stage to another. But it is opposite in this case. In case of the thrombopoiesis, the formation of platelets, we have the megakaryoblast, and you can see here the size of the megakaryoblast is 15 to 20 micrometers. Okay, and this megakaryocyte contains a big nucleus. It contains a big nucleus, and there is the basophilic cytoplasm, and nucleus contains multiple nucleoli. Okay, because it has to be involved in the formation of different proteins. We will talk about that. It has to be involved in the formation of different contents of the granules of the platelets. Fine. So megakaryoblast, 15 to 20 micrometers. Now this megakaryoblast will differentiate into another type of cell that we call as promegakaryocyte. Promegakaryocyte before the megakaryocyte. So promegakaryocyte, we can see that the size will increase from 20 to 30 micrometers. So that means size of the promegakaryocyte will be increased as compared to the megakaryoblast. Okay, now the cytoplasm is still basophilic, but not as basophilic as the megakaryoblast. There are multiple nucleoli present there, and there will not be the round nucleus. There can be oval shaped nucleus, and this nucleus is slightly shifted to one side. So it is not in the center, it is not round, but it is oval shaped and slightly shifted to one side, and the cytoplasm is slightly less basophilic as compared to the megakaryoblast. Now, this promegakaryocyte will go for the process of endomitosis. Now, what is mitosis? Mitosis is the cell division, but cell division involves the process of the DNA replication, and at the end there is cytokinesis. Now, in this case, what is endomitosis is that there will be the replication of DNA. DNA will go from 2n to 4n to 8n to 16n to 32n to about 64n. So DNA will be replicated many times, but there will not be the process of cytokinesis. It will remain one cell, but there will be the replication of DNA inside the cell. So we can see, while going from promegakaryocyte to another type of cell, it will be differentiated into megakaryocyte. Now, megakaryocyte mein we can see that there are many lobes of the nuclei. Okay, in many lobes of the nucleus, and it is because we have gone for the process of endomitosis. Okay, now the cytoplasm is not basophilic. Now there are different granules present inside this cell that is called as megakaryocyte. When we talk about the size of the cell, it can be 35 to 60 micrometers. Okay, in some books it is written as that it can be up to 90 micrometers. So you can imagine how giant this cell is, how big this cell is, this megakaryocyte. Now, megakaryocyte now will form the cellular extensions. Okay, it will form these cellular or cytoplasmic extensions, and we all know that it is present in the bone marrow. Okay, it is present in the bone marrow. For example, here is the bone marrow, and here we have this megakaryocyte, for example. Inside the bone marrow, we have the sinusoidal capillaries. What are the sinusoidal capillaries? They have the gaps present in between the endothelial cells. For example, here is one endothelial cell, here is another endothelial cell, here is another endothelial cell, and these endothelial cells are making this capillary. So this capillary is the sinusoidal capillary. Now what will happen, that these extensions will increase in the size. These extensions will increase in the size, and these extensions will increase and they will go through these gaps. Okay, these sinusoidal gaps. Now these extensions are called as proplatelets. So that means that these extensions are going to give rise to our platelets, the cellular fragments. Now, once these proplatelets, these cellular extensions go through the sinusoidal gaps in the sinusoidal capillaries, because of the force of the blood that is going through these capillaries, there will be budding off of these proplatelets and they will form these fragments. We call them as platelets. Okay, now we can see here in the previous slide, you can see here we have the megakaryoblast, and from megakaryoblast we have the promegakaryocyte. Promegakaryocyte will go for the process of endomitosis, and there will be the formation of megakaryocyte. And megakaryocyte have these cellular extensions, and it is multinucleated, or there are different many lobes of the nucleus, and it has these cellular extensions. They will be increased in the length, and these cellular extensions are called as proplatelets. Then because of the force of the blood in the sinusoidal capillaries, these proplatelets will bud off and they will form these fragments. We call them as platelets. Now we will talk about the regulation of this thrombopoiesis, regulation of the formation of platelets. The main important thing that is regulating this formation of platelet is called as thrombopoietin. Okay, that is called as thrombopoietin, and this thrombopoietin is released from liver and kidneys. But the main site, unlike the erythropoietin, the main site is not kidneys, it is the liver. Okay, so liver being the main site for the secretion of this thrombopoietin, and thrombopoietin will go to the bone marrow and stimulate the process of thrombopoiesis, stimulate the process of the formation of platelets. The other things are interleukins, most importantly the interleukin 6, and there is another thing, granulocyte monocyte colony stimulating factor. Okay, but these are not having that major role in the formation of platelets. The major role is played by the thrombopoietin, but they definitely assist the thrombopoietin, or they also help us in the formation of platelets, that is thrombopoiesis. One more important thing is that platelets follow the feedback mechanism. Okay, if there is low platelets in the blood, that will stimulate the liver to secrete more thrombopoietin. More thrombopoietin, that will go to the bone marrow, increase the production of platelets. And if there is increased platelet count in the blood, what will happen? That will inhibit the liver to form the thrombopoietin. That will not, so that means enough thrombopoietin will not be available to stimulate the bone marrow for the process of thrombopoiesis. That will decrease the platelet production. Okay, so that means it is actually following that feedback mechanism. Now let's talk about the structure of platelets now. Okay, now structure of platelets, as I've already told you, that they are not the complete cells, they are the cell fragments, and they are made after the budding off from the giant cell that is the megakaryocyte. So once these cellular extensions bud off, those proplatelets bud off, okay, only the cytoplasm and the other cell organelles present in the cytoplasm will be the part of these platelets, not the nucleus, because nucleus is in the center of the megakaryocyte. Okay, so what we have to see is that these platelets will be anucleated or non-nucleated cells or cell fragments. There will not be the nucleus present there. Okay. Now, one more important thing is that चाहे ये सेल फ्रेगमेंट्स ही क्यों ना है, चाहे ये पूरे सेल्स नहीं है, बट स्टिल दीज प्लेटलेट्स हैव गॉट some developed जो है cellular structures. The first thing is it has got the highly developed cytoskeletal system. Okay, it has got the highly developed, it has got the microtubules and microfilaments. Okay, we will talk about that in a little more detail in the lecture. Then it has got the spatial canalicular system. Okay, spatial canalicular system, we will talk about that. Canalicular system is the canaliculi are the invaginations in the cell membrane of the platelets. We will talk about that. Then it also has got the developed membrane systems, that also we will talk about in the lecture. Now let's first talk about the cell membrane. Okay, cell membrane of the platelets. Now it is definitely the phospholipid bilayer, like the cell membrane of the other cells. This platelet is the phospholipid bilayer, but there is important thing that the exterior coat, this cell membrane jo hai, it is covered by an exterior coat, and that is called as glycocalyx. Okay, that is called as glycocalyx. And this glycocalyx jo hai, it contains, it is, this glycocalyx is made up of glycoproteins. Okay, and these glycoproteins can include the glycoprotein receptors. Okay, and it is also made up of glycolipids. Actually jo glycocalyx hai, that is the carbohydrates in the cell membrane, but carbohydrates are not free. They are either in the combination of lipids forming the glycolipids, or in the combination of glycoproteins in the combination of proteins forming the glycoproteins. Okay, and those glycoproteins are very, very important, because glycoproteins on the cell membrane of the platelets form the receptors, glycoprotein receptors, that are very, very important for the functioning of the platelets. We will also talk about that. Now, this platelet has the negative charge on the surface. Okay, it has the negative charge on the surface, and that negative charge is specially given by a sugar molecule that is called as sialic acid. Okay, and this sialic acid jo hai, it gives the negative charge to the surface of the platelets. Now what is the significance of this negative charge? It will repel the platelets from each other. It will prevent the aggregation of the platelets, or it will prevent the sticking of platelets to each other in the normal circulation, because in the normal circulation we don't want the platelets to get stick to each other. They will form the clot in the normal circulation. So in order to prevent that sticking of platelets to each other in the normal circulation, we have the sialic acid giving the negative charge to the platelets. Okay, that is very important. Iske baad we have in the membrane we have the membrane glycoproteins, and these glycoproteins are very, very important, because for the proper and normal functioning of the platelets, these glycoproteins have got a tremendous role in the functions of platelets. The first glycoprotein is glycoprotein 1b9. Okay, 1b9. Suppose, let me show you here, we have a cell membrane here, sorry, we have a blood vessel here, and there is the injury to the blood vessel, there is a puncture to the blood vessel. Now what will happen? There will be collagen that will be exposed. There will be collagen that will be exposed, and here, for example, is the endothelial cell that is damaged. Okay, now this damaged endothelial cell will secrete a chemical, and that chemical is called as von Willebrand factor. Okay, that chemical is called as von Willebrand factor, and that von Willebrand factor will act as a bridge between the exposed collagen and the platelets. Okay, now once this von Willebrand factor will be secreted here, it will help the platelets to stick here. Okay, to adhere here, we call that as platelet adhesion. Now, platelet adhesion is the sticking of platelets on a non-platelet surface, because von Willebrand factor is not a platelet. It will help the platelets to stick on the non-platelet surface, and to stick with the von Willebrand factor, we have one important glycoprotein on the membrane of the platelets, that is glycoprotein 1b9. The second is glycoprotein 2b3a. Okay, now once the platelets are adhering, we have completed the process of platelet adhesion. It will be followed by the platelet aggregation. That means sticking of the platelets on a platelet surface. Now one platelet will stick on the one Willebrand factor, and it will be activated. It will attract more platelets, and those platelets will now stick on the platelet surface. Now for the platelet to platelet sticking, there is an important जो है receptors, and those glycoproteins are glycoprotein 2b3a. Okay, now suppose one Willebrand factor is not there. Okay, one Willebrand factor is not there. There can be the direct adhesion of the platelets on the exposed collagen, and that is in that we have for that we have a glycoprotein, that is glycoprotein 1a 2a. Okay, now these are the receptors that help the platelet adhesion directly on the exposed collagen. So we have three. The first is glycoprotein 1b9. It helps in the platelet adhesion with the 1 Willebrand factor. Glycoprotein 2b3a, platelet aggregation, platelet with the another platelet. We have glycoprotein 1a2a. It helps in the platelet adhesion directly on the exposed collagen. Now, the next thing is cytoskeletal system. Now, cytoskeletal system, mein, what did I tell you? We have the microtubules and microfilaments. Now, look here. We have the microtubules here, and this microtubules, jo hai, these microtubules actually microtubules, they actually जो है they help in maintaining the shape of the platelets. Platelets are not spherical, they are the disc shaped, these platelets, and to maintain that disc shape there is an important role of the cytoskeleton, and that is called as the microtubules. Okay, it provides that structural integrity to the platelets. The second thing is microfilaments, and microfilaments mein there are three important filaments. We have the actin, we have myosin, and we have thrombosthenin. Thrombosthenin. Fine. So actin, myosin, and thrombosthenin. So what is the role of these microfilaments? They help in changing the shape of the platelets, because platelets during activation has to change their shape, and to change their shape there will be the role of these microfilaments, actin and myosin. Now, like the muscle cells, these platelets can contract, because you can see here actin and myosin is present there inside the platelets. So they are the contractile cells. So that is the role of the microfilaments there inside the platelets. Okay, now one more important thing is that we talk about the cellular system, and cellular system we will talk about two things. One is the open canalicular system, second is the dense tubular system. And open canalicular system, you can see here, these are the invaginations in the cell membrane, and these invaginations going deep into the cell. Okay, there are cell membrane mein there are invaginations, and these invaginations are making the canals, they are making the tunnel shaped structures, and these canals are called as open canalicular system. Okay, now what are they meant for? Actually जो इस platelet के अंदर granules present होते हैं, and those granules में जो composition होता है, उन granules के जो contents होते हैं, they have to be secreted from the platelets, and they will be secreted in these canals, and they will be then secreted out. Okay, so these canals act as the tunnels for the secretion of the granules that are present inside the platelets. The second thing is that there is deep tubular system. Now, what are these deep tubular systems? These are actually the sarcoplasmic reticulum, or endoplasm, not sarcoplasmic, the endoplasmic reticulum. Now, endoplasmic reticulum, most importantly, the smooth endoplasmic reticulum, and smooth endoplasmic reticulum, we already know that it has the very, very important function of storage of calcium. Okay, and that calcium is stored in these in this deep tubular system, and it is actually the endoplasmic reticulum. Okay, and this endoplasmic reticulum is not highly developed endoplasmic reticulum, but it is still there, and it stores the calcium that is involved, for example, during the contractile process, and that calcium is stored there in the endoplasmic reticulum. Now, moving forward, we have now the granules that are present inside the platelets. We have two types of granules, actually three types. One that is present in every cell, that is lysosomes, but two other types of granules we have inside the platelets. One is the alpha granules, and second is the dense granules, or dense, we call that as dense bodies. Okay, now when we talk about the alpha granules are 50 to 80 granules in one platelet. Okay, and they are about 200 nanometers in diameter. And when we talk about the dense granules, they are just 3 to 8 granules per platelet, and they have the diameter of 20 to 30 nanometers. So you can see that alpha granules, they are maximum in number and they are larger in diameter. And when we talk about the dense granules, they are fewer in number and they have with the small diameter. Now, when we talk about the contents of these granules, we have the alpha granules. They actually contain the protein substances. Okay, they contain the protein substances. For example, they contain the adhesive proteins. Okay, adhesive proteins, fibrinogen that helps in the platelet aggregation, von Willebrand factor, fibronectin, thrombospondin, they help in the platelet adhesion. Okay, platelet adhesion during the formation of the platelet plug. The second thing is the growth factors, and these growth factors are very, very important for the healing process. Okay, once there is the injury, after the platelet plug formation, there has to be the healing process to heal that injured area. And for that, we have the growth factors, most importantly, the platelet derived growth factor. This is very, very important. And these are responsible for that healing process. The third thing is the coagulation factors. And these coagulation factors, for example, fibrinogen, factor 5, factor 9. अब ये coagulation factors जो है, these are included in the clotting factors, the 13 clotting factors that are very, very important for the process of coagulation. Then when we talk about the dense bodies, they contain the non-protein substances. For example, the ADP that attracts the platelets, for example, the serotonin, and this serotonin is a vasoconstrictor because at the site of injury, we need the vasoconstriction to decrease the blood flow to that area. Okay, so serotonin is also present in these dense granules. And then we definitely have the calcium, that is one of the clotting factors that help in the process of coagulation. Now, the life history, it is formed in the bone marrow and it is destroyed in the spleen, mainly in the spleen, but it can be destroyed there in the by the macrophages of liver as well. Okay, so these platelets जो है, they remain alive for, or they remain in the circulation for 8 to 12 days. Okay, they remain in the circulation for 8 to 12 days, and then they lose that discoid shape. They lose that disc shape, and after that they get stuck in the, for example, in the small capillaries of the spleen, and they get destroyed there. Just like we have seen in the RBCs, they lose the biconcave shape. Just like that, these platelets, they lose the disc shape and they get stuck there, and then the macrophages come and they destroy them. Okay, then these platelets जो है, ये platelets जो है, ये liver में भी ऐसे ही जो है destroy हो सकते हैं. But one more important mechanism we have to talk about is destruction in the liver via the Ashwell-Morel receptors. And these Ashwell-Morel receptors are, for example, मैंने अभी आपको कहा था that there is sialic acid giving the negative charge to the cell. And once the cell becomes old, the sialic acid goes away. The sialic acid is not there. And that sialic acid is not there, that means we cannot prevent the aggregation of platelets. So we need to destroy them. So once the sialic acid is not there, it actually exposes the galactose that is present under the sialic acid. And there are the receptors in the liver for that galactose, and those receptors are called as Ashwell-Morel receptors. Now, Ashwell-Morel receptors are like, oh, there is no sialic acid, there is galactose, come, I will grab it, and this platelet will be grabbed there, it will get stuck there, and then it will be taken into the liver and will be destroyed there. Okay, so that is the destruction of platelets. Now, what are the functions of platelets? Platelets have the very, very important function in hemostasis. Hemostasis is stopping of bleeding. Okay. Now, hemostasis is completed in two steps: formation of the primary hemostatic plug and formation of the secondary hemostatic plug. Now, these platelets have the role in both. Now, primary hemostatic plug may actually, there will be the process of platelet adhesion and aggregation. Now, what will happen? मैंने अभी आपको दिखाया था, this is the blood vessel and there is the injury. There will be exposed collagen, there will be von Willebrand factor released here, and platelets will come and get stick here. Okay, platelets will stick, that will be platelet adhesion. And after that, these platelets will be activated and they will release certain chemicals. We call that as, after activation there is release reaction, and those chemicals will attract more platelets and those platelets will come and stick to these platelets here. That is called as platelet aggregation. And then more and more platelets will be called by these platelets, and those platelets will come and they will come, and this process will go on, and there will be a plug of platelets formed, and there will be a clump of platelets formed, and that is called as primary hemostatic plug. This is not a clot yet. Okay, the clot is the secondary hemostatic plug. There will be all that cascade reaction, and the end result of that will be converting the fibrinogen into fibrin. Then that fibrin will come and make a network here on these platelets, and now it will become a clot. That is called as secondary hemostasis. Now, in secondary hemostasis, there is the role of platelets. For example, these platelets provide the surface for that coagulation process. That is the role of platelets. Now, platelets contain, abhi humne dekha tha, the platelets contain fibrinogen, the platelets contain factor 4, the platelets contain factor 9, the platelets contain calcium. All these are very, very important for the secondary hemostasis. So, platelets definitely have the role in the secondary hemostasis as well. Now, another thing is clot retraction. Clot retraction is the contraction of clot. Okay. Now, for example, the platelet plug is formed, secondary platelet plug, secondary hemostatic plug is formed, clot is now formed. Now, there will be the retraction of the clot. There will be the contraction of the clot. Actually, these platelets, with the help of that actin myosin, they will contract. And that contraction will help in, for example, if that contraction is done, this contraction of this clot will pull the two edges of the injury close to each other, and that will promote the healing process. The second thing is that once there will be clot retraction, and that clot retraction will decrease the clot size, that will promote the blood flow inside the blood vessel. So this is called as the clot retraction, and there is definitely the role of platelets. Now, there is thrombolysis. Okay, thrombolysis jo hai, or we can say platelets control the fibrinolysis. Okay, once the clot is formed, now when that clot has to be lysed or destroyed, that is controlled by the platelets itself, because platelets know that the healing process has been done, and while releasing certain chemicals जो है ये इस fibrinolysis process को भी control करते हैं. The third thing is the storage and transport. Now, platelets synthesize, secrete, and transport many chemical substances that we have seen in the form of alpha granules and dense granules. There are certain important things that are present in the alpha granules and the dense granules. So that is another function. Then another function is phagocytosis. Now, because I have already told you that these platelets has the tendency to change their shape. So, like the white blood cells, the neutrophils and macrophages, these platelets are also can do the phagocytosis, but not as big phagocytic cells as the white blood cells. They can phagocytose smaller molecules like immune complexes and the viruses. So definitely the platelets have the property of phagocytosis. Then the vascular growth. Vascular growth, मैंने आपको अभी थोड़ी देर पहले बताया था that in the alpha granules we have the growth factors. Most importantly, the platelet derived growth factor. And this platelet derived growth factor जो है, it is very, very important for the vascular growth. Okay, vascular growth or for the healing process. दैट इंजर्ड साइट में जो है हीलिंग प्रोसेस होनी है. एंड द अदर जो वहां पे हमने देखे थे ग्रोथ फैक्टर्स, दे कैन प्रमोट द एंजियोजेनेसिस एज वेल. सो दीज ग्रोथ फैक्टर्स जो है या वेस्कुलर ग्रोथ में जो ग्रोथ फैक्टर्स चाहिए होंगे, दे आर प्रेजेंट इन द प्लेटलेट्स. सो प्लेटलेट्स आर इन्वॉल्व इन द वेस्कुलर ग्रोथ एज वेल. Now let's talk about the normal count and the variation in the normal count of the platelets. Now the normal count is 1.5 to 4.5 lakhs per cubic millimeter of the blood. Okay, sorry, 1.5 to 4 or 4.5 lakhs per cubic millimeter of the blood. Okay, now if it goes less than 1.5 lakh per cubic millimeter, that is called as the thrombocytopenia. So thrombocytopenia is the variation and it is the decrease in the platelet count less than 1.5 lakhs per cubic millimeter. Okay, now there is something called as critical count, and this critical count is when the platelet count is below 50,000 per millimeter square, per cubic millimeter. Okay, it is less than 50,000 जहां पे जो है ब्लीडिंग जो है ब्लीडिंग ज्यादा होने के चांसेस है, ब्लीडिंग जो है रुकने के चांसेस बहुत कम है, बिकॉज प्लेटलेट्स हैज टू प्ले एन इंपॉर्टेंट रोल देयर. एंड इफ द प्लेटलेट काउंट इज लेस देन 50,000 एंड दैट इज कॉल्ड एज द क्रिटिकल काउंट. Now, what are the conditions that will decrease the platelet count? The first thing that you have to understand is one autoimmune disorder that is called as idiopathic thrombocytopenic purpura. Okay, isme, as it is autoimmune disorder, so auto antibodies will be formed, and those auto antibodies will start destroying these platelets. And what will happen is there will be the decreased count of these platelets, so that will lead to thrombocytopenia. Then there is aplastic anemia. Aplastic anemia, that means bone marrow is depressed and that will produce less platelets, so that will lead to decreased platelet count. Then hypersplenism. Now what happens is hypersplenism में क्या होता है कि about 70% एक्चुअली नॉर्मली क्या होता है कि जो टोटल प्लेटलेट्स होंगे उसमें से 70% ऑफ द प्लेटलेट्स आर इन द सर्कुलेशन एंड 30% जो है दे आर शिफ्टेड to the spleen. They are stored in the spleen, we can say. And if there is hypersplenism, for example splenomegaly, there is enlargement of the spleen. There is enlargement of spleen, the spleen will store more and more platelets, more than 30%. In some cases it can go up to 90% of the platelets get stored in the spleen. So what will happen? The circulating platelets here, that number will decrease and that will be thrombocytopenia. Okay, and definitely because of these जो है cytotoxic chemotherapy and radiations, there will be depression in the bone marrow that will lead to the decreased production of platelets and that will lead to thrombocytopenia. Okay, now the last slide we have that there is the thrombocytosis. अब thrombocytosis जो है इसमें it is the increased platelet count. It is more than 4.5 lakhs per cubic millimeter. Now what are the conditions? For example, there is polycythemia vera and chronic myeloid leukemia. So what will happen? The bone marrow will be super active, and bone marrow will be super active. It will be producing all the types of blood cells. It will be producing all the types of blood cells more in number. So definitely there is the increased production of platelets as well. And if there is increased production of platelets, that will lead to thrombocytosis. Okay, now iron deficiency anemia. If there is anemia, there is that जो है feedback mechanism में क्या होगा? There will be secretion of erythropoietin, and that erythropoietin जो है कभी-कभी क्रॉस होता है, एंड दैट एरिथ्रोपोइटिन जो है नॉट ओनली प्रमोट्स द बोन मैरो और स्टिमुलेट्स द बोन मैरो फॉर द प्रोडक्शन ऑफ आरबीसीज, दैट विल स्टिमुलेट द बोन मैरो फॉर द प्रोडक्शन ऑफ these platelets as well, and that will lead to increased platelet count. Okay, then splenectomy, अभी मैंने आपको कहा that spleen जो है it stores 30% of the platelets inside. And if there is splenectomy, that means removal of spleen, that means spleen is no longer storing these platelets, so all the platelets are there in the circulation, that will increase the count of the platelets. Then chronic infection, surgery, or acute hemorrhage. Most importantly, the chronic infection. And this chronic infection, what will happen is there will be inflammatory cytokines or inflammatory markers will be released there. मैंने आपको शुरू में कहा था that the regulation of thrombopoiesis में हमने देखा thrombopoietin था, and there was something called as interleukin 6, and that interleukin 6 will be released or secreted in case of chronic infections and inflammations. So that interleukin 6 will definitely promote the process of thrombopoiesis, and that will lead to the increased this platelet count, that will lead to the thrombocytosis. So that is all for today, my dear viewers. Thank you so much. See you in the next lecture.